What we wanted to do was figure out exactly how many tumors out there have somatic mutations within the homologous recombination pathway. We've known for quite some time that BRCA mutations are important in terms of your risk of developing cancer during your lifetime, but also how you respond to specific therapies, notably platinum, chemotherapy, and PARP inhibitors. BRCA mutation carriers actually perform much better, and there's also evidence that that's true with folks with somatic mutations as well. And there's kind of growing evidence that there's many additional DNA repair proteins that might possibly have similar clinical significance. But we haven't really well defined those proteins yet, and we certainly don't know how prevalent they are or if patients with these mutations will respond similarly to, again, platinum chemotherapy and PARP inhibitors. So we wanted to just kind of broadly look out there across all tumor types to see how often we could identify these kind of newer mutations. We ultimately had access to over 53,000 tumors. We partnered with Caris Life Sciences, which is a commercial company that can perform extended molecular profiling for any patient across the United States that has obviously a tumor biopsy. So in partnering with them, we got to analyze quite a large number, certainly the largest that anyone's ever done before. And ultimately, we were able to identify that 13% of the tumors that we tested had one of these mutations. And we looked quite broadly. There's kind of ongoing debate about which mutations should be considered, which genes are of importance. But we ultimately studied about 20 different genes.
Going for all tumor types, finding all mutations, that's surely just like a pin at infinite number of dartboards.
Yes. But that's also what's kind of exciting is, a lot of the work right now has been focused on breast cancer, ovarian cancer, now kind of extending into prostate, pancreatic, a little bit into head and neck. And we really wanted to see, hey, is this more of a issue all across the board, and are there even more patients that could potentially benefit from specific therapies or, potentially benefit from more intensive screening? That's certainly a long way away, but just to kind of start that more broader search. There's a lot of clinical trials ongoing right now that are starting to pull in kind of these bigger groups of mutations, coined homologous recombination deficient tumors. Most of these trials are looking at PARP inhibitors. Some are now starting to draw in immunotherapy, but we still have a ways to go, but that's the ultimate goal.
How does this, you think, influence the possible design of future trials, having this kind of these more, I would say narrow targets, but it seems to be just about everything everywhere, but having at least some sense of where to start looking and possibly even coming up with new drug designs, new targets that way?
Right. I think the first step, and what we're also trying to do at Georgetown, is designing trials where you're kind of shooting broadly and capturing all these different either germline mutation carriers or with evidence of the mutation within the tumor itself, with the hopes that giving them these targeted therapies like PARP inhibition will work, but we don't really know for sure yet that more broadly this is going to work beyond BRCA. It might take a few years to really identify additional genes that are as impactful of a player as BRCA. Even PALB2, we don't have all the data on that gene either.
Well, I suppose you could try and sum this up with maybe a conclusion, a summary, a final message.
Sure. So I think the biggest kind of take-home points from our study was that homologous recombination mutations are quite common. We identified mutations in 13% of the 53,000 plus tumors evaluated. Other groups have also published on homologous recombination deficiency and have had similar percentages or even higher. So it's certainly something important. And then what was unique to our study too is we saw these mutations very broadly. So you could have a non-small cell lung cancer patient, even though now they have lots of treatment options, but potentially, even though you wouldn't think of a PARP inhibitor for a lung cancer patient, maybe if they have evidence of homologous recombination deficiency, they might benefit from that treatment as well. And then just to kind of give folks a quick look at what we found in terms of the specific genes. So we evaluated for PTEN. There's a lot of controversy if that's truly a gene that's important in the homologous recombination pathway, but it was our most commonly mutated gene in 5.8%, and then BRCA2 and BRCA1 followed at about 2.5% to 3%, And then ATM and PALB2 were our next most commonly mutated genes. And then in terms of which cancer lineages were the most commonly affected, ovarian was the most commonly affected lineage, which we all know from previous literature. For us, we got mutation rates in 14.1% of all the tumors tested. Surprisingly, bladder was next at 9.7%, and then breast followed at 8%. So that was also kind of fitting with current literature.
It just sounds like such an awful amount of space to take up, just like the sheer physical storage of that much information.
But that is the future, trying to get these clouds with all of our information that, if we could all collaborate a little bit better in these clouds, because there's a lot of information out there that we're all doing separately, and if we can get it together, maybe we can start answering these questions a little bit better.